if you're here online or here in person, thank you guys for coming. This is the first in-person or hybrid version of our talks that we're able to do, which is very exciting that we can have people join in both ways, including our speaker. So welcome everybody to the meeting of the New York City Neighborhood Naturalists group, which is part of the New York City EcoFlora project. My name is Lydia Paradiso. I'm a Ph.D. student at the New York Botanical Garden and a graduate fellow with the New York City EcoFlora project. NYBG first created the EcoFlora project back in 2017 with a grant from the Institute of Museum and Library Services. The EcoFlora is a community science project that has three main goals: to investigate urban ecosystems and urbanization, to support open source biodiversity data, and to increase understanding and appreciation of plant life among city dwellers. To date, over 30,000 observers have made over 800,000 observations of plants and their biotic partners in New York City. You can explore those observations on our project page in iNaturalist, which I'll link after we finish this intro. I'll also link our YouTube page, which has recordings of our past talks, and we'll also have a recording of this talk if you have to leave early or you would like to share this with anyone afterwards. For the people that are online, as we go, you can enter questions into the chat, or you can do so at the end, and we'll be able to read those and have our speaker answer them. People in person, if you guys have questions, we'll be able to give you the mic at the end to ask that. And now I will turn it over to Tomi for our land acknowledgement and intro. Thanks, everybody.
Hi, everyone. My name is Tomi Barrett, and I am the project manager of the EcoFlora project. I'd like to begin this seminar with a land acknowledgement by recognizing the original people of this land, the Lenape, as well as their deep connection to the Lenape-Hoking homeland. As an organization dedicated to advocating for the more than human world, we must acknowledge those who are original stewards of this land. We believe in the importance of acknowledging the settler colonial genocide perpetrated against the indigenous communities, as well as the resilience of the Lenape, who still today continue to resist erasure. All right, for our introduction, Sophie Breitbart is an urban ecologist and evolutionary biologist investigating how urbanization influences the monarch butterfly's favorite botanical partner, the common milkweed. After completing her BA in biology at Wesleyan University in 2016, she worked as an environmental consultant assessing lake water quality and designing engineering plan sets for dam removals. She began her Ph.D. in Ecology and Evolutionary Biology at the University of Toronto in 2018. Her current research focuses on a garden she planted, which contains 1,000 milkweed plants grown from seeds collected from an urban, suburban, and rural areas. She has observed the milkweed over the past 4 years to answer this question: do plants exhibit observable differences that suggest that urban and rural plants are genetically different from one another? Next, she will use genomic tools to further investigate how city life influences common milkweed on a molecular level. In her free time, Sophie likes to instruct coding workshops and plan her next Zabars order. All right, Sophie.
All right. Hi, everyone. My name is Sophie Breitbart. I am really excited to be speaking to you today on behalf of the EcoFlora project. So I'm a PhD candidate at the University of Toronto, and today we'll be exploring how urbanization, how cities impact the ecology and evolution of plants. But before we start talking about cities, I want you to think about a time when cities as we know them now did not exist. You know, when there was no skyscrapers, there was no traffic, there was no Broadway, really none of that. So rewind 400 years ago, approximately, to the early 1600s. And so I want you to think about when New York looked like this, when it was this lush green island that was covered with deciduous forests and streams and hills and valleys, when you might see beavers running around collecting material for their dams, or you might see bears foraging for food in the woods. You might even see whales swimming in the nearby waters. And you would also see the Lenape indigenous peoples who, for thousands of years, hunted and farmed and fished on the land that they called Mannahatta. And we know a lot about the species that were living here because of this really wonderful initiative called the Mannahatta Project, which was started by a landscape ecologist named Eric Sanderson. So has anyone here heard of the Mannahatta Project before? All right, we've got one hand, great. So to fill you in, long story short, Dr. Sanderson and his team were able to predict which species likely lived throughout Mannahatta on a very small spatial scale, based on some incredible work involving gathering historical information about the area, using historical maps, as well as modern spatial data about the area, and also information about the species that were thought to be living in that area at the time. And then they use these incredible cutting-edge statistical tools to predict where each species could have lived on Mannahatta based on, number one, the resources that they needed to survive, but also their close relationships with other species. And so, for example, if they said, okay, someone wrote about seeing a beaver in Mannahatta hundreds of years ago, they would think, so we know that the beaver needs water to build its dams, and so it needs to live near a stream, but we also know that it needs to eat. And so it can only live in an area if its prey are nearby, for example. And so all in all, there were multiple products that came out of this project, but one of them is this incredible interactive map explaining which species likely lived throughout modern day Manhattan on a block by block basis. And so I do encourage you to check it out after the talk. It's really interesting. So what happens to these species when we build the largest city in the United States on top of an ecosystem that was as biodiverse as Yellowstone National Park? Well, we know several ways that their environments can change. So for example, we know that cities tend to be warmer than the more rural or natural areas surrounding them due to something called the urban heat island effect. And so I think the EcoFlora project has put on some incredible talks in the past, and I'm curious from listening to those talks, or maybe just by walking throughout the city, can anyone here shout out some ways that urbanization changes the environment? We've already got that cities tend to be warmer than the areas surrounding them. Pollution, yes. Say that one more time. Yes, lots of impermeable surfaces, less permeable surfaces, exactly. Fragmentation, exactly. All right, I think you guys, you really hit the nail on the head. So we've already heard that cities tend to have higher amounts of pollution of all kinds. We've got road salt, smog, noise pollution, light pollution, the list goes on and on. There's also less green space in cities due to increased habitat fragmentation. So when you do find a green space, it's probably fragmented, so it's cut in size than what it used to be, and they tend to be isolated from one another, so a little bit further apart than they used to be. We also see changes in the species that are found living in cities through this phenomenon called biotic filtering. And so this means that some species that need, for example, very large territories or very specific conditions in order to survive probably aren't going to be found living in urban environments. But on the flip side, we see that some species are commonly introduced into cities or they independently move in themselves, like sparrows. You see sparrows living in cities all over the world, and they're really able to make it work. Okay, so now that we've covered some ways that urbanization changes the landscape, let's think about how species respond to these changes. So one thing we know is that oftentimes urban populations, which are just groups of individuals of the same species. That's what I mean by populations. Urban populations have different phenotypes compared to non-urban populations. And by phenotype, I'm just talking about an observable characteristic. So for example, shell pattern is a phenotype, leaf color, heart rate, anything like that. So for example, researchers have found that this plant called Virginia pepperweed was larger in urban areas as opposed to rural areas. And so this example illustrates an important concept called phenotypic divergence. So just the phenotype is diverging among these groups. We also know that urbanization can impact reproductive success or a species' ability to reproduce. And so again, these researchers found that the urban Virginia pepperweed had higher reproductive success than the rural Virginia pepperweed, since those urban plants produced more seeds. Great. Okay, so now we've thought about how urbanization changes the landscape and some ways that species respond to those changes on an individual basis. But now I want you to take a second and think back to what I mentioned about how Dr. Sanderson and his team made their Mannahatta map. So first they thought about the species that were thought to be living in Mannahatta, but then they also thought about the relationships among those species. They thought about the species that they ate, the species that they tried to avoid getting eaten by, the species that they pollinated, the species that they competed with, lots of species interactions. And these networks of species interactions are incredibly complex. They're huge, and they can involve thousands of species. And in order for these ecosystems to be properly functioning, in order for them to work well, they should be balanced. And so again, what happens to these interactions when we build the largest city in the US on top of this incredibly biodiverse ecosystem? The short answer is that we really don't know much, even now. And so to find out, I focused in on asking how urbanization impacted the interactions between plants and pollinators specifically. And I had good reason to think that I might find something here for a few reasons. And so first, we already know that urbanization impacts both of these groups individually. So for example, we know that the communities of plants and pollinators are often different in cities versus rural areas. We've also seen that in some urban areas, the urban plants are flowering earlier than their pollinators can keep up. So the pollinators are sort of running late in a way. And so we wondered if those changing phenological patterns for the plants could mean that those plants aren't being pollinated as well. And third, scientists have also found that urban air pollution can reduce pollination by decreasing the number of visits that those pollinators make to the flowers. And so again, all in all, we had reason to believe that urbanization might be impacting these interactions between plants and pollinators. And so to dig deeper, I asked this question. How does the reproductive success of a native plant and the community structure of its pollinators vary along an urbanization gradient? And so by community structure of its pollinators, I'm just talking about if you were to go out somewhere in the Bronx and you see a plant being pollinated by wasps and bees and butterflies, I would ask you, how many pollinators do you see? How many species do you see? Are there some species that are rare and others that are dominant? Things like that. And so again, the idea behind this question is to see if urbanization is impacting the interactions between a plant and its pollinators, and if there seems to be consequences for the plant's reproduction as a result, because this plant relies on pollinators for its reproduction. And the plant in these plant-pollinator interactions is ninwish, as the Ojibwe indigenous peoples call it. You might also know it as common milkweed or Asclepias syriaca. So common milkweed is a perennial, meaning that it can live for many years, often decades. It's a plant that you can find living in cities, suburbs, rural areas. It's really good at just growing where it's planted or wherever it finds itself. It's really important for conservation since it's the main host of the monarch butterfly, but it's also pollinated by at least 20 other insects. And fun fact, I understand that monarchs and milkweeds were the first New York Botanical Garden EcoQuest targets. So it's got some clout there too. And lastly, common milkweed is found throughout much of North America, especially the Northeast and the Midwest, where it's considered a native perennial. And so I performed this research in the Greater Toronto area of Ontario, Canada, which some people like to think of as the New York of Canada. And so to start answering my question, the first thing that I did was to draw a straight line on a map from Toronto city center out towards the countryside, called a transect. And that transect was around 50 miles long. And then within that transect, I found 80 sites with common milkweed. And I will say that we found a lot of these sites just by driving around and keeping our eyes peeled. But also, I found many of these sites by going on iNaturalist the day before and seeing what observers had posted to the site. And so that's just one way that I use the data that EcoFlora provides. So the data is really being used by scientists, including myself. And so since this transect was on Toronto's urbanization gradient, I was able to use each site's distance from the city center as a proxy for urbanization. And so after I found a sample site, I visited it twice per year for 2 years. And per site, I visited, or I sampled up to 5 plants per site. And so in July, I started by surveying the amount of pollen that had been removed from the milkweed flowers in order to get an estimate of male fitness, which is just a component of reproductive success. And with milkweed, it's really easy to do this because each milkweed flower has 5 pollen sacs or pollinaria, which you can see in the red circle in the left picture. So you can see that that little black dot is present. And in the right picture, you can see that it's been removed, probably by a pollinator, just like this one that we found scampering around on a leaf of a plant that we were sampling. And so you can see that the pollinaria is tangled around its legs. Then again, we went back in September to record a few more metrics. So first, I recorded the number of peduncles, and each peduncle signifies an inflorescence or a flower cluster. And so that acted as our estimate of reproductive effort per plant. And second, we also counted the number of follicles per plant, or those seed pods that you'll see open up, especially in the fall. And then to study how the pollinator community structure changed along that urbanization gradient, we visited these same milkweed populations in 2019 only, so 1 year. And what we did was we pulled out our lawn chairs. We put them around 3 meters away from up to 3 plants per site. And then we stared at those plants with binoculars so that we could identify the pollinators that visited these plants within a 5-minute period. Okay, so what did I find? Well, before I show you my results, I'm just going to walk you through a typical figure. So on the x-axis, which is down here, we've got distance to city center, spanning from very urban on the left side to very rural on the right side. And on the y-axis, we've got our response variable. So for us, that's going to be, for example, the amount of pollen that had been removed per flower. And so really what we see here is all over the board. So the first thing that we see is that urbanization negatively affected pollen removal, though the amount varied per year. So first we saw that there was around 10 to 20% of pollen removed per flower in the city versus around 40% at the rural end of that transect. We saw that the impacts of urbanization on the number of follicles or seed pods really depended on the year. Urbanization had a positive effect on fruit set or the number of follicles per inflorescence, but there was really no impact for inflorescences on its own. So if we turn our attention to the pollinators now, the first thing that we see is that urbanization negatively impacted these pollinators in terms of their abundance or how many of them that we saw. So overall, we saw around 60% fewer pollinators per plant in the city versus in the rural countryside. However, we saw more species of pollinators in the city as compared to the more rural parts, but we didn't see any impacts for species diversity. And so to conclude, we found that even though those urban plants were pollinated less often, they still experienced higher fruit set than the rural plants. So they're producing lots of offspring. They're not experiencing lower fitness. They're okay. And this is happening even though urbanization seems to be impacting the pollinator community in lots of ways, various impacts. And so I think this finding is especially interesting because oftentimes plants and pollinators respond to urbanization in parallel, in very similar ways, but that's really not what we found here. And I think this is a surprising finding, and it's pretty hopeful that maybe these urban species interactions are a little bit more resilient than we thought. Okay, so up until now, I've been talking about urbanization like it's the simple metric where you go out to a site and you say, this is 20 miles from the city center, and so that's how urban it is. And I will say that for this transect specifically, the one that I studied, that metric is trustworthy, and that my colleagues have found that distance from the city center strongly correlates with other metrics like canopy cover and impervious surface coverage. Impervious surface just meaning like the amount of buildings and parking lots and roads that you see within that area. But I think if we really want to capture the complexity of urban areas, then we need to consider the landscape itself. And so here I'm just showing you a bird's eye view of Toronto, and you can see all the different types of land cover that you might see in a typical urban area. And again, I think it just helps to give a sense of how varied cities can really be. They're not just covered with parking lots and roads all over the place. They're really mixed up. And so as of now, we still don't really have a great understanding of how the landscape itself impacts phenotypic divergence among these urban and rural populations. And so my second research question focuses on asking how a specific landscape feature of urban areas might impact phenotypic divergence in common milkweed. And that landscape feature is the urban corridor. And I chose to focus on corridors specifically because we know that they can influence evolution in non-urban settings. And so what is a corridor? How does it influence evolution? So a corridor is generally thought of as a long, relatively connected strip of habitat. And so on the top panel here, you can see an example of an urban corridor that we found in downtown Toronto. And you can see it looks really different from the bottom aerial, the bottom picture, which represents a more typical view of an urban area. Okay, so now that we know what corridors are, how can they impact evolution? So I think when many of us hear evolution, we think about adaptation and animals camouflaging into their backgrounds and maybe Darwin's finches and their beaks. And while I think that natural selection is an incredibly interesting aspect of evolution, it's actually just one of 4 processes, with the other 3 being gene flow, genetic drift, and mutation. And so the 2 processes that are most relevant to our study of corridors are going to be gene flow and genetic drift, though natural selection and mutation are certainly still relevant. I just won't talk about them quite as much. Okay, so what is gene flow? So gene flow refers to the movement of genetic material among populations. And so for a plant like common milkweed, this could look like when a pollinator takes pollen from a plant in one population, brings it to another population, inserts it into a flower there. That flower goes on to produce seeds, and oftentimes that second population will end up with higher genetic diversity than it had before, which is often a good thing, but not always. And so oftentimes what we see with urbanization is that it restricts the movement of these pollinators among these urban populations. And so sometimes the populations are very small and isolated, and the pollinators just can't fly far enough to connect them. The air pollution in cities that I mentioned earlier might be masking the sense of the flowers, so they might be having a harder time finding them. But all in all, there are lots of ways that city life can impact pollinator movement. But the point here is that we have reason to believe that urbanization might impact evolution by decreasing pollinator movement among these populations and ultimately reducing gene flow between these urban and rural milkweed populations. But in non-urban settings, corridors have also been shown to influence gene flow, and they frequently do this by increasing gene flow amongst these otherwise isolated patches of green space. And this is really important for our study of urban milkweed because, again, when we see an urban milkweed population, it's often small and very isolated from the other ones around it due to habitat fragmentation. And so this really made us wonder if corridors might help facilitate gene flow in urban areas, just like how they do in non-urban settings. And on the other hand, corridors can also impact another aspect of evolution, which is genetic drift. So genetic drift is a process that randomly changes the genetic composition of a population's gene pool pretty much due to random chance or sort of sampling error from generation to generation. And oftentimes that results in a loss of genetic diversity. And again, losing genetic diversity in this case is often a bad thing, not always, but it happens more frequently in those small and isolated populations, often what we see in cities. And so again, in non-urban settings, corridors have been shown to connect these isolated patches, and as a result, they can reduce the effects of genetic drift. And so we thought maybe these corridors in cities could impact genetic drift as well. And so this all leads me to my second question. How does proximity to urban corridors influence the reproductive success of common milkweed and the community structure of its pollinators? And so to answer this question, I took my initial transect and I split it into 3 sub-transects. And so the first 2 were parallel, they're on the same length, though the southern sub-transect followed green corridors like trails and rights of way, and the northern sub-transect went just straight through the city grid, right through that urban matrix, while the third sub-transect extended from the suburbs into the rural parts. And so here's an example of a sample site that we found in the urban non-corridor sub-transect, where you see just a few milkweed next to a really big parking lot. And in contrast, here's a site that we found along the urban corridor sub-transect, which is just within a trail. And so for the second question, I only looked at the populations that were located within either of those 2 urban sub-transects. So I did not look at the rural sites here, but I used the same sampling data that I had collected as I explained earlier. And so what did I find here? Okay, so first thing that we found was that the plants living along that urban green corridor produced more inflorescences, more flower clusters in the 2 years that we sampled. And we can see that in this graph by the 2 red-orange lines, these warmer lines over here, that are just generally higher up compared to these blue, darker lines down here. When it came to pollen removal, it was really complicated. It depended not only on the distance from the city center, the year, but it also varied by subtransect as well. So it was complicated. We didn't see any differences in the number of follicles or seed pods produced in the green corridor versus the non-green corridor, and it was the same for fruit set or follicles per inflorescence. When we turn to the pollinators, what we saw is that there were actually fewer pollinator species present along that green corridor. So we saw around 20% fewer species, lower species richness along that corridor. We also found that the diversity of those pollinator species was around 75% lower along that corridor compared to the non-corridor. But we didn't see any differences for pollinator abundance. And so to wrap up here, when we found that urban corridors did influence plant reproductive success, the results were generally positive, even though aspects of pollinator community structure were affected by urban corridors sometimes. And when they were, they tended to be negatively impacted. And so although species interactions might be impacted by these urban green corridors, the milkweed are not really suffering in terms of reproductive success. They're doing okay. And I think this is a really interesting result, and we could unpack this for a long time in terms of the relevance for urban planning and the utility of green corridors in cities. But I do want to mention that other studies have shown that green corridors are really helpful for lots of species, including pollinators and plants. And like networks of species interactions are incredibly complex. And so, maybe this suggests that green corridors impact plants in a positive way, just maybe not through their pollinators, I'm not sure. But I think we've really got to repeat this study in lots of cities with lots of other plant and pollinator groups around the world to really get an idea of what's going on here. Okay, so by this point, I hope that I've shown you that urbanization sometimes impacts species through which they respond by changing their phenotype. So we see phenotypic divergence occurring. And we've seen this not only in plants, we've seen this in many other species as well. But for the most part, we still don't really understand where this phenotypic divergence comes from. And so there are three main factors here that could have contributed to this phenotypic divergence. So the first factor is the environment. And the environment can shape an organism's phenotype through non-genetic means within a generation. And this illustrates a really important concept called phenotypic plasticity. And so to give you an example, let's say that we have a group of plants and they're all genetically identical. And we put half of that group of plants into the city, the other half into the countryside, and we watch what happens over the lifetime of those plants, just one generation. So we see that the urban plants end up being taller than the rural plants. And because we know that those plants were genetically identical, we can attribute those different phenotypes to phenotypic plasticity, or to them altering their phenotype through physiological changes. Phenotypic divergence can also be due to differences in genetic makeup or genotype. And so that illustrates genetic divergence. And so an example of when we might see this is, again, we take the same group of genetically identical plants, we split them up, put them into similar or the same conditions, and then over many generations, we ultimately see that one group looks very different from the other group. And because we kept their environments the same, we can conclude that those phenotypic differences that we see are instead due to differences in genotype. So maybe, they've accumulated different mutations over time, something like that. And finally, the third factor is the intersection of these two phenomena, called a genotype by environment interaction, but we don't have to worry about that right now. And so again, researchers are racking up lots of examples of phenotypic divergence occurring between these urban and rural populations, but only a few of those researchers have asked, what is the source of that phenotypic divergence? And so that's where my research comes in. And so what I'm interested in for this question is asking whether that phenotypic variation that we observed earlier in my first chapter is consistent with genetic divergence as a result of evolution, as opposed to the other factors. And so basically, I want to know, are the urban milkweed populations genetically different from the rural populations? And it's plausible to think that we might find evolution happening in cities, because we've seen it happen in many other species around the world, sometimes within our lifetimes. It can happen really quickly. We didn't expect to see this when we first started, opening our eyes to this phenomenon, but it can actually happen. Urban evolution can happen very quickly and repeatedly in species around the world. And so to get at this question, I am testing the hypothesis that urbanization drives genetic divergence in phenotypic traits among populations of common milkweed. So here my first step was to collect milkweed seeds from those same populations that I sampled earlier. And so I germinated about 1,000 of these seeds. They were taken from urban, rural, suburban environments, and then I completely randomized them into a common garden experiment. And if you're not sure what a common garden experiment is, that's totally fine. Just think about how I mentioned that there are those three main factors that contribute to phenotype. So the purpose of a common garden experiment is to help us figure out, for example, how much a plant's height is due to its genetics, its genes, the nature part, and the nurture part, so the environment. Because it's never nature or nurture, it's often both of them. And the common garden experiment does this by minimizing the environmental factors that contribute to phenotype, by raising lots of individuals from very different environments in the same conditions. And so, for example, the way that this works is that you can collect lots of squirrels from an elevational gradient, or whatever species you want from a temperature gradient. For me, I took lots of milkweed seeds from an urbanization gradient. And then I planted them all in a single garden, the same conditions, so that each seed grows up with the same environmental conditions. And so when we assess how tall, for example, a plant is in a common garden, that trait should predominantly reflect the plant's genetic makeup rather than its environment. And so on one hand, if we see that the urban plants are all much shorter than the rural plants in that common garden, then that would tell us that there's a high amount of genetic divergence between those urban and rural populations for plant height as a result of evolution. But on the other hand, if let's say the plants are all very similar in terms of height, then that would show us that there is very low genetic divergence for height. And so again, I took those 1,000 seedlings, planted them in a common garden experiment. Here's what that common garden looked like. Those seeds came from 52 populations along that gradient. And then for 4 years, I started monitoring over 24 phenotypic traits. And so these traits fell into 3 main categories, being plant defense, growth and development, and reproduction. Okay, so did we find evidence that urbanization has influenced genetic divergence in phenotypic traits in this plant? Well, the first thing that we found was that urbanization was positively associated with the concentration of these toxic secondary metabolites, these chemicals in the plant leaf tissue called cardenolides. And cardenolides, even though they can be tolerated by monarchs, it generally prevents herbivores from chewing, for example, on the plant leaves. We also found that urbanization was positively associated with the probability that a plant would be attacked by herbivores, specifically before flowering began. And so if you're scratching your head wondering why would seemingly the best defended plants also be at the highest risk for herbivory, we were also wondering that too. But before we could really dive into that, I needed to do one more thing, which was to address the fact that I had treated these over 24 traits like they were independent, which they were not. And so, for example, we know that plant height before flowering is going to be very well correlated with plant height after flowering. We know that plant height will probably go far in predicting how many inflorescences a plant will produce, things like that. And so when I accounted for this non-independence, I used 2 ways to do it. So I used a false discovery rate test. I also did a multivariate analysis. Basically, I found that those 2 results that I just found were likely false positives and that they showed up as meaningful due to random chance. And so taking into account all these 3 lines of questioning, it looks like when these plants do exhibit phenotypic divergence among these urban and rural areas like we saw initially, that divergence is not due to evolution, and instead it's primarily due to phenotypic plasticity and potentially these G by E interactions. And we found very similar results, qualitatively identical results when we brought in the corridor. So we didn't see much evidence for genetic divergence between the corridor plants and the non-corridor plants as well. And so, many other people have detected rapid urban evolution in plants and many other species around the world, but we have not found that yet in common milkweed. And so what might be driving these results? We had lots of questions. Well, one idea, one potential reason why we might not have found evolution in this plant comes from the life history traits. And so what I mean by that is milkweed is not only a perennial, but it's also clonal. And so if you were to go out and find a field of milkweed somewhere in the Bronx, it's possible that you could be looking at just a few genetically unique individuals that have been growing for decades. And so these populations might have only also been exposed to urbanization for a few generations because Toronto is a very young city. It's less than 250 years old. But, regardless, there are lots of reasons why we might not have found genetic divergence, and that's really an interesting result in itself. And so for my next project, I'm going to try to figure out, why this is. And so basically, my experiment showed me that in general, these urban and rural populations are pretty similar to one another genetically. And so the next thing that I've done is to sequence the DNA of milkweed from around 100 sites throughout Toronto, the urban and rural areas, and to try to figure out which processes might have led to these patterns arising. And so there are lots of potential processes that might have led to these patterns. So for example, we know that people really like to plant milkweed in their gardens to help the monarchs, of course. And so we know that people like to go to nurseries. And so maybe what happened was that they planted these seeds from nurseries, which the seeds are very genetically similar. They planted them in their yards. Those plants went on to produce their own seeds. And since milkweed is wind dispersed, they blew throughout the city. And we ended up sampling lots of relatives of those plants. And so that would make them very genetically similar. Another option of what might be going on here is that maybe there's just really high gene flow between these populations. Maybe these urban pollinators don't have as much trouble going from milkweed population to milkweed population as we thought. And so that could also lead to those same patterns that we've observed. Great. Okay, so to wrap this up, I hope that I've shown you that urban environments can affect plants, they can affect pollinators, and the interactions between them. But, to go back to my title, I noted that there are these two-way streets in urban evolutionary ecology. And I think to fully appreciate the ways that urbanization can impact the ecology and evolution of the species that live inside these cities, we've got to realize that this is only one small part of that whole big picture. And in reality, there are these endless feedback loops connecting these plants and pollinators. So for example, we know that milkweed's toxic cardenolides, those chemicals that help prevent herbivores from eating them, they're not only present in the plant tissue, they're present also in the nectar, as well as many other parts of the plant. And so that can impact the pollinators that come and feed on the nectar and end up pollinating the milkweed. And so raising the cardenolide levels in the plant might be helpful to deter herbivory. So for example, scientists have seen that monarchs laid fewer eggs on milkweed that had higher levels of cardenolides in its nectar. But if the plant raises those cardenolide levels too much, it can make the pollinators sick, and that is not what the plant wants. The plant needs pollination through these insects. And so in this way, we've got a two-way street where the urban environments that impact pollinators, those pollinators go on to impact the plants, but then the plants impact the pollinators, and it just never stops. And so this is an example of a dynamic feedback, as scientists like to call them. And so again, this is only one small part of this entire story. So urban environments are affecting way more than just plants and pollinators. They are affecting so many species. And so, for example, city life could be impacting the brown snake, which impacts its food source, the salamander. That goes on to affect the snake again. It can affect the snake's predators like a blue jay. It just keeps going on and on and on. And so urban environments are totally reshaping these networks of species interactions. And I think when you really sit down and think about it, it's just so totally mind-boggling. And you might not even be thinking about the evolution part of this. It's so wide-ranging. And so I don't expect New York to look like Manhattan again anytime soon. 400 years ago, that lush, beautiful green island. But I think it's still very clear that our cities are full of this incredible biodiversity and these species interactions that we can't even fathom how they're being affected by city life yet. But a big step towards understanding how city life is affecting those interactions is by getting on the ground and seeing what's going on. And again, that's what the EcoFlora project, that's what you all are doing. And so I really commend you for your efforts. And I am honestly really excited to see what we all find out together. And so I'll just stop here. I'd really love to thank the organizers, Tomi and Lydia and Lisa, for inviting me and helping make my visit happen. This is really like one of the coolest talks I've ever given. So thank you so much. I'd also love to thank my advisors, Mike Johnson and Helen Wagner, as well as both of my labs at the University of Toronto, the incredible team at the Koffler Scientific Reserve, which is where I did my common garden experiment, and you all for listening. And I would be happy to take your questions. So thank you so much.
Thank you all for joining, and everybody in person, thank you all extra. We're opening up our Zoom for questions now. So if you have a question and you are on Zoom, please type it in the chat. And then Lydia will read it out loud. It looks like we've got some coming in now. And then thank you again, Sophie, so much for coming.
All right, let's see if I'm going to be a disembodied voice back here for now. We have some questions on the Zoom, and then if anybody in person has questions, can take my mic. Okay, first question we have from Kelsey. Was there any difference in percentage of visiting native and introduced pollinator species between the urban and rural environments?
That's a great question. We didn't dive into that so much, but we did notice differences, for example, in the amount of honeybees that were pollinating these plants in urban and rural environments. I don't exactly remember the patterns, but we did notice different numbers of honeybees. And so if you'd like to reach out to me, I'd be happy to get you that information if you'd like, Kelsey.
Awesome. All right, we have also some questions from Amy, and I have some questions related to this too. So her first question was about the cardinalides, which she asks, are the cardinalides inducible by herbivory? And then I have a related question to that, which is how did you measure the cardinalide content?
Yeah, so the cardinalides are induced by herbivory. As I understand it, they can have a baseline concentration, but they can also be triggered by herbivores. So yes. And so how did we measure cardinalide concentrations? Well, the short answer is that we sent them off to our wonderful collaborator, Anurag Agarwal, who is known for his research on monarchs and milkweeds. And he used, I think it was a mass spec, which like mass spectrophotometer, something like that. And so again, if you'd like more information on that, I'd be happy to get it to you, but I didn't do that work myself. He just kindly generated the data for me.
This thing about having so many collaborators, you can do a lot of different kinds of things that people have expertise in. All right, another question we have from Amy about the pollinators. Did you distinguish effective pollinators from just flower visitors?
Another great question. No, we counted a pollinator as an insect that landed on a flower and looked like it was pollinating, but we didn't distinguish specifically if an insect just seemed to be landing and like immediately left and did not seem to pollinate. Interestingly enough, we found that some red soldier beetles, which are not commonly thought of as pollinators of common milkweed, they just like walk a lot over the flowers, but we noticed that they had pollinaria wrapped around their legs. And so if they had ever deposited those pollinaria back into the stigmatic slits of a flower, they would be considered pollinators. And so we saw that. It was pretty interesting.
And I guess as someone who doesn't study the pollinator part, I think it probably could be difficult to fully, know what's really going on at that small scale if you're just observing. But okay, and the third question we have from Amy, along the urban transects, did you measure distance to gardens?
We did not. That would have been a really great way to incorporate the heterogeneity, or like the way, just how much the urban landscape is varied into our analyses. And yeah, sometimes people plant milkweed in their gardens to attract pollinators because it's so great for pollinators. And so they will oftentimes pollinate their crops as well. So yeah, urban gardens can certainly impact the patterns that we saw, but we didn't explicitly include them in our analyses.
Just highlights how complicated all of these systems can be and how different they are. Like you said, if you do it in different cities, every city has a different layout and different makeup that would affect.
Exactly, different histories, and the land use of cities has changed a lot over the years for different areas around the world. The historical impacts, the legacy impacts. Cities are so dynamic and they're really hard to characterize with just a distance from city center, for example, but we're getting better at the ways that we're quantifying them, but they're these incredible mixtures of ecology and the human side of things as well.
All right, and we got one more question in the chat here. Did you measure a difference in species diversity and pollinators between the 3 different subtransects that you had?
Yep, so we didn't measure species diversity. No, actually we did. So yes, part of our analyses involved understanding how the species diversity of those pollinators varied both along that urbanization gradient, but also between those 2 urban corridor and non-corridor subtransects. So we did measure species diversity as well.
All right. Is there anyone, do you have a question? Okay, perfect. I'll bring this down.
So the question was, were we able to specifically count the number of wasps that were present in the diversity of those wasp species, right, between the urban corridor and non-corridor subtransect? Yes, so we, to the best of our ability, I wasn't actually the one who did this work. It was done by a very motivated undergraduate named Albert Tomshishin, who is an author, a co-author on the paper that we just published about this research. So to the best of his ability, he characterized the pollinators down to the species level if he could, but if he couldn't do that, he would characterize by genus or family, the best that he could. And sometimes they were categorized as morphospecies. So, this wasp was different from this wasp, and that's how the best we could do in terms of identifying those species. I don't know exactly if the diversity of those wasps differed among the corridor and non-corridor subtransects, or if the wasp species diversity changed along the urbanization gradient as well. But I do know that we saw wasps. They are frequently seen on milkweed. So thank you.
Hi, thank you so much. This was very cool to see how you have sort of attacked this question. But I wanted to ask, so do you think it would be useful, or what could it tell you if you did the opposite of a common garden experiment and did transplant experiments? Do you think that could tell you something more about?
Yeah, that's a great question. So if I understand correctly, you're asking, what if instead of a common garden experiment, we did a reciprocal transplant experiment, right? So with a reciprocal transplant experiment, that helps you detect local adaptation. which is a really interesting question in itself. And that's sort of what I'm getting at with some other research that I didn't talk about today through another method. But with the reciprocal transplant, you take, for example, lots of urban milkweed and you grow them up in a very rural environment. And then you do the same thing where you take lots of urban milk or rural milkweed and grow them up in an urban environment. And then you can test how the fitness differs among those urban and rural plants to get a sense of if, for example, urban milkweed have adapted to their urban environments. And some other people in my lab are looking into that, and they have found examples of adaptation to cities, especially in white clover. That's a really hot species right now for that. And so I do think there's a lot of value in doing a reciprocal transplant experiment. I didn't do it. I did think about it briefly, but milkweed, they take a lot of work to get going. And, clover is a little bit easier to handle. And that would have meant finding a huge plot in a big urban area. And I think it would have been a challenge. It's certainly possible, but it's easier to ask those questions in species that don't need quite as much room, like white clover, for example. So definitely worth asking. And if you're interested, I would love to see what you find.
Hey, thank you again for your great talk. I have a question related to the genetic differences between urban and rural populations. Did you also compare it with completely different populations from another state or from, I don't know, that would be very far from the Toronto populations, whether it is related to this, to the city and the areas around, or if it's as like in the whole species, the genetic diversity between population is very low.
Yeah, that's a great question. We did not grow any species or any individuals outside of that gradient. And so, yeah, the species or the individuals that we used, they might have, this comes in with the next question that I'm going to be asking, but they might be technically part of the same genetic population with its own gene pool. We might also find that there are different genetic clusters within our urban area, suburban area. But no, we did not. But I think that if we were to do this again, that could be interesting to add individuals from outside of our Greater Toronto area sampling area.
Okay, I have one more question about this, the sequencing. You don't have to go into too much technical detail, but what kind of sequencing are you doing? And how will you plan to analyze those results?
So I think big picture, I'm using a technique called genotype by sequencing, which will, in the end, it's giving us, it's very different from whole genome sequencing, which is where you get the entire genetic code for each individual. And so for GBS, instead, we're getting around 1% to 2% of the entire genome, but it can still tell us a lot. That's a lot of data. Yeah, was there a second part to your question?
That was it. I was just
Okay.
curious, because especially like that we're doing in our graduate studies, et cetera, some kind of things like that. So I was like, okay, that's what I would have guessed, something like that.
Other people in my lab have used whole genome sequencing to study urban evolution in different plants. So you can find out even more from that.
And especially when you have a lot of samples that you want to be able to get stuff with, how much money you have all comes into play.
Absolutely.
All right, Sophie, we have one more from the chat from Amy. Given the clonal habit, do you have any ideas how old milkweeds can be?
No, I mean, one thing that we did was that we refrained from sampling milkweed that we knew had been planted. But the milkweed that we sampled otherwise, we really don't know how old it was or if it was a clone. We tried to sample individuals that were at least 5 meters apart. And so that could help us theoretically sample different genetic individuals, different genets, but that was our best guess. Without really digging down and looking at the root system, we wouldn't know.
Great. And then for me, so with the species diversity of pollinators between the two transects, did you compare those both to a rural species diversity of pollinators, or was it just along the transects, sort of the end of them are sort of rural, right?
Yeah, we didn't involve the rural part there. We were just trying to understand in the city, does a green corridor seem to impact these other aspects of plant reproduction and in the pollinator community structure? So we didn't involve the rural part of the subtransect in those questions.
Great, thank you.
All right, last chance. I think we got all our questions exhausted. Thanks to everyone for coming in person and virtually. I'm glad we were able to offer both options. Thanks so much, Sophie, for coming and giving us this talk, presenting to us the research that you've done so far for your thesis. And showing us that it's not as cut and dry, that it's not just, oh, urbanization is just across the board bad for plants, that it's much more complex than that. And there's lots of cool ways to look at it.
Absolutely. Yeah, we are here to stay, I think. And so we need to keep living with the biodiversity all around us and find ways to cohabitate. So urbanization is not inherently bad. There can be some really bright spots. So thank you for hosting me. This has been a great time. And thank you all for listening.
And stay tuned. We'll have more hybrid events, or at the very least Zoom ones. Yeah, so thanks again, everyone, for coming.